Hello, I'm Poe Bronson. I'm Managing Director of IndieBio San Francisco, and this morning is the online portion of the demo day for IndieBio Batch 14. Our 10 companies have been working very hard for the last 6 months, culminated in the videos capturing their magic that you're about to see. The total show will last about 45 minutes, and you'll be done and out of here in under an hour. Mohan, do you want to give some instructions for investors?
Yeah, you'll find information about each of our companies. Look below. You'll have decks as well as calendar links with the CEOs so you can make your appointments. It is a hybrid demo day. We have an in-person portion of our demo day at 4 o'clock for all investors. Please come and visit us at the new location at Dogpatch to see our companies.
And we'll run with the show. We'll see you after the show. With VIA, I'm so excited about how big it can be and how fast it can get there. Their 3 molecules can decarbonize 100 billion gallons a year of the fuel market. And now, while a lot of investors, they hear the word biofuels and they can't see straight for a moment, but they forget that ethanol scaled fast from 2005 to 2011, in just 6 years. It became 10% of all of US auto fuel. And we can run the same playbook with a lot of the same players and the same feedstocks.
Here's what jet fuel has to do inside a jet engine. First, your plane is at 30,000 feet. It's about minus 50 degrees outside, so your fuel can't freeze up. Then the fuel is pressurized to 23 atmospheres. It's preheated above its flash point, but below its auto ignition point. And when it combusts, it flashes above 3,000 degrees Fahrenheit. So you do not want to get this wrong. And we do not want to redesign the engine to work with a new decarbonized fuel. I'm Louis Dutel. I've been an entrepreneur and an operator in oil and gas for 20 years. Our CFO Brent was CEO of a global tech company that connected deep water wells on the seafloor to the surface, operating for decades in the middle of the ocean. Our CSO Phil has 40 years in molecular biology and wrote the first textbook on biomanufacturing. We have the team to do this job. It's pretty simple why VIA will win. Let's go back to class. For jet fuel, you need hydrocarbon molecules of 7 to 9 carbons, cycloalkanes. Our competitors start with ethanol, which is just 2 carbons, or syngas, which is just 1 carbon, and use lots of expensive steps to gradually add up to 8 carbons where it's finally a fuel. At VIA, we are the shortcut. From almost the same system that makes ethanol, from the same Iowa corn or Queensland sugar, using the same type of yeast that makes 8 carbon fuel that can drop straight into a gas tank, we cut out all the steps between. See this double line in the benzene ring? This means we're an aromatic. So in the bioreactor, our fuels volatilize and are pulled off the headspace continuously. This changes everything. Our reactors can run longer, more continuously, and in turn, we can reach economics that ethanol never could. We have a full stack of partners. In Iowa, where the corn is grown, we're working with Iowa State University and Biomade to replace ethanol. In Queensland, Australia, where they grow sugarcane, we have a biorefiner with 25,000-liter continuous bioreactors ready to turn on. We have super major oil and gas partners, and we even have airlines on board, ready to take off. We can do this today. We can sell every bit of fuel we grow today and not have to wait anymore. Our fuels can start decarbonizing jets, trucks, even existing cars today.
Sim is one of these founders that just has amazing founder market fit. He was quite literally born into a textiles manufacturing dynasty. And so he learned from childhood about the ins and outs of this this industry. And it really shows in the type of team that he's assembled to do this hard thing and the technology that they're trying to make work. And maybe more importantly, the like no kidding metrics that they're trying to hit because they know what the standards are. And it's a team of veterans who have the tenacity of a really young, hungry startup. They're quite literally at this moment driving from New York to North Carolina to get a pilot run done. And they're gonna transport 100 pounds worth of this stuff over to Tennessee afterwards to complete the pilot.
Our story starts with wartime parachutes, stocking riots, and this guy. In 1935, DuPont's Wallace Carruthers unleashed a world-changing technology, nylon, the world's first synthetic fabric. A polymer that came from oil but mimicked the pliability, feel, and strength of silk. Nylon's invention kicked off a 90-year explosion of synthetic fiber development that has left our modern society completely and utterly dependent on synthetics, even for clothes. Clothes, something humans have been making from natural fibers for over 35,000 years. At Bloom, we're not trying to turn back time, we're plotting a new way forward. One that exploits the best smart materials from nature, aka proteins, and marries them with the best of industrial fiber manufacturing, better known as melt extrusion and melt spinning. Look, 70% of the textiles market is built on melt extrusion and melt spinning. $150 billion worth of polyester fibers are made every year using these technologies. If that doesn't scream scale, I don't know what does. I'm a third-generation textiles man, and I know that if we're going to make a dent in the textile industry, we have to integrate with existing infrastructure. It keeps our CapEx and OpEx low, and well, that's just smart business.
You've probably heard this story before. A young startup comes to market with a new protein-based product that's going to replace plastic. But you get under the hood and you learn that they're not using melt-based machines. And in this industry, if we're not using melt, you're toast. We're different. We're meeting the fiber industry right on their factory floors, feeding their melt extruders and melt spinners with our proprietary K pellets.
Keratin proteins, grown and thrown away by the billions of tons every year, which we have converted into relaxed, moldable, all-natural thermoplastic. They're a stable, easily processed melts that are manageable at 250 degrees Celsius, just like polyester. Today we're extruding, drawing, and spinning fibers, but we can't get to market fast enough. Luxury brands, sportswear brands, and even automotive conglomerates are asking us, begging us, how soon we can deliver our samples. To meet our demand in the next year, we will be producing 1,000 kilograms per month, and at that scale, we'll be competitive with recycled polyester. I'm Sim Galati, co-founder and CEO of Bloom Labs. Together with farmers, brands, mills, and scientists, we are transforming the textile industry towards a more circular future. Join us.
Laura Shea from Tipping Point. Wow, what a pleasure it's been to get to know this amazing scientist. Comes out of the chromatin biology lab in the country, Geeta Narlikar at UCSF. Always been impressed with her scientific knowledge since I met her for the first time. But what's more impressive is that she has scaled her assay, a unique assay that the world has never seen, which you'll hear about in just a second. Scaled it in a way that she's made more chromatin in her lab than probably all the labs combined. Not only that, she has made selective small molecule hits against a rare pediatric disease when other companies with tens of millions of dollars invested have not been able to produce. All this in 3 months.
Aberrant chromatin is the starting point of many diseases. It is best characterized in cancers, making it a high-value target for oncology. Every cell in your body has the same DNA sequence. So how is it that a brain cell is a brain cell and a skin cell is a skin cell? It's how the DNA is packaged. Certain genes are packaged such that they're inaccessible and genes are turned off, and other genes are open so they're turned on. Just like DNA packaging is important for normal cell identity, it's when that DNA packaging goes awry that you get disease. Conventionally, a lot of people have tried to go after DNA packaging by targeting single proteins or interfaces between two proteins. But what we know is that in disease, a lot of times it's the chromatin network or the DNA packaging network that really is problematic. I'm Laura, the CEO and co-founder of Tipping Point Biosciences. My co-founder, Dr. Geeta Narlikar, who's a professor at UCSF, is a world leader in chromatin biology. With our expertise, we have invented a new type of screening platform. We can identify molecules that specifically modulate the disease chromatin network without affecting the healthy ones in a test tube. Our platform is working. We are identifying an entirely new class of molecules that work in a different way. And our initial hits are showing efficacy in patient cells without being toxic. We're taking a really different approach at how we're doing drug discovery. I want you to take out all the assumptions for targeted-based drug discovery. Because we know that it's chromatin networks that are really causing the disease, we need to redefine what the target is. And the target is a chromatin network. How do you go about drugging this chromatin network? You have to create an assay that enables you to really understand how these interactions are changing. We can recapitulate a network that is known in disease, and then we can find small molecules that modulate these interactions and break only the bad and not the good interactions. The first disease indication that we're focusing on is a pediatric brainstem tumor called diffuse intrinsic pontine glioblastoma, or DIPG, because it's the best proof of concept for our platform. In the last 4 months, we have scaled this assay so that we can screen for 10,000 molecules now. We know that there are many diseases driven by aberrant chromatin networks. DIPG is just the tip of the iceberg, and we are planning to go after many other diseases, including other cancers, neurodegenerative diseases, and also developmental disorders. Biology is complex, and reducing it to single interactions or interfaces has only taken it so far. At Tipping Point, we are not just building a new path to the same destination, we're unlocking a new destination entirely.
The thing about Alkali Labs is that these founders, Luis and Jacob, there's more than meets the eye with them. They're two super smart synthetic biologists training under Jay Kiesling, the OG synthetic biology lab at Berkeley. And you have no doubt that they really get the science that they're trying to build into the technology. You would also not expect that they would have any idea how to sell a partnership to an oil and gas operator. And these guys have taken that task super seriously. They've dug in, they've rolled their sleeves up, they've gotten their hands dirty, and they just got back from a 4 or 5 day trip down on the bayou of Louisiana, visiting with independent oil and gas operators, talking to them, understanding their problems, and building those relationships that are going to be the foundations of commercial partnerships.
Okay, hot take. The oil and gas industry is the key to electrification.
No, it's actually true. Here's why. Every year, potentially billions of dollars worth of lithium are pumped and unceremoniously dumped by the oil and gas industry in America.
Hold up, let me rewind. See, it's hard to mine lithium in the US. Either we don't have enough rocks for it, or we don't have the lakes thick with lithium that can be cheaply evaporated by the thin desert air. But what we do have in the US is lots and lots of lithium-containing brine deep underground. It contains between a few hundred parts per million lithium down to below 50 parts per million.
Megatons worth. Tens of billions of dollars worth.
So knowing that, you may automatically think, well, hey, let's find the highest concentration brines that we can and figure out a way to mine those. And a lot of people would agree with you. In fact, most would. So back to that hot take, Alkali Labs is going to mine lithium from brines as low as 50 parts per million. And we're going to do it with brine that the oil and gas industry is already bringing up to the surface.
Yep, it's true. For every barrel of oil, between 3 and 20 barrels of mineral-rich brines are produced, to the tune of over 20 billion barrels of this trash every year.
These brines are either too dilute or too contaminated with oil to even attempt state-of-the-art direct lithium extraction.
We're turning the clean, high ppm brine paradigm on its head. Because why start new brine drilling projects when we could get megatons of lithium from the stuff coming out of oil operations today? I'm Luis Valencia.
And I'm Jacob Roberts, and we're Alkali Labs.
We're developing a technology to take ready-to-use waste brines and turn it into refined lithium. Right now, we're connecting directly with oil and gas operators across America who are leaving megatons of lithium on the table, just hiding in plain sight in their produced water.
Our microbial miners are miniature vacuums for lithium, engineered microbes that can process and gobble up the mineral content in brines as dilute as 50 ppm. The other guys require brines with 400 ppm or more in order to make their numbers work.
They also need fresh wells, chemical pre-treatments, high-performance resins, membranes. Every step's just another cost driver. And they definitely can't work in oil-contaminated brines. Our workhorses don't just survive in produced water, they're thriving in these freshly collected, untreated oil-filled brines. See?
Our distributed plug-and-play technology can integrate with the on-site operations of junior operators and super majors alike. We can work with your brines straight up. We only need to partner with 10 medium-sized US oil fields in order to start generating hundreds of millions of battery-grade lithium carbonate revenue per year.
With Alkali Labs, we're bringing lithium production to America to secure our electric future.
Join us.
It's not every day you run into a team of hustlers, and when you do, it's a great feeling. This next team is a team of hustlers. Vika, Revital, they're out there on a flight. They're out there at conferences. They're out there selling the startup. These are two industry insiders who come from fashion. They've seen the underbelly of this industry, and they know what it takes to make this industry sustainable. And that's been a thrill to see with this startup. They're learning directly from the market. May it be talking to car manufacturers, what parts they want to get rid of in their cars, or talking to machine manufacturers, what machines they can rent. These two people really embody what price discovery, market discovery is all about. And you'll see that in this next video.
Hi, I'm Victoria Kanam, co-founder at Refresh Global. I met Revital, my co-founder, at a sustainable fashion event I organized. We were both in the fashion space, and we both felt the need to find a solution for the fashion problem. We had been in enough warehouses, showrooms, and high-end fashion shows, knowing well what happens behind the scenes when the show is over. We looked at the building blocks of modern-day textile waste. Increasingly, natural components like cotton are blended with synthetic fat polyester. We thought to ourselves, why fight the variability of textile waste? Why not work with it? Refresh Global has a solution for that. We sourced this waste from the city of Kfar Saba in Israel as part of a pilot run. We ended up upcycling 60% of the materials. What you see on your screen is a synthetic nonwoven fiber. This fiber is the raw material for resin pulp, a product with many use cases. It can be needle punched to go into couches and cushions. It can be thermopressed and molded into shapes that will become cupboards or car trunks. You can also airlay it to make insulation boards for offices. Our process starts by separating these building blocks. The synthetic components are separated and upcycled into the nonwovens you saw above. The organic components, on the other hand, go through our proprietary enzymatic hydrolysis protocol, a process that breaks down the cellulose in textiles and refines it into our hero product, nanocellulose, a high-value product with use cases spanning industries from food to batteries to materials. Our tech lead, Dr. Taki Shalev, is spearheading the optimization of our enzymes in partnership with VTT in Finland, one of Europe's finest research institutes. We have preliminary data that puts us on track to get high-yield nanocellulose and patent our enzyme blend and methods. We learned directly from the market, securing a paid pilot with Volkswagen from the nonwoven pressed into the car trunk. We showed fashion giants how we can take that stock from molds and make it into hangers. Each day has been an adventure. Touring factories such as Andretz La Roche to learn about machines we can rent or use turnkey in our process. We visited Zoex, our first partner, one of the largest textile redistributors in Europe, where we will pilot our first of many in-situ microfactories with machines already available in the market. Refresh Global is our way to make a difference. We give the industry a refresh, from trash to trusted.
I get a lot of oncology companies come by my desk all the time, a lot of them proposing to go after solid tumors in different ways. And what I love about Karma is that their proprietary asset, their LNP that they designed in-house, doesn't go after the tumor itself, it goes after the macrophages that end up being half the tumor anyways. And what's special about that is now you get to leverage the exponential nature of the immune system. You get to go in, create this chain reaction effect, you get to create this virtuous cycle in which you actually kill the tumor. So it's a really interesting mishmash of immuno-oncology and in vivo cell therapy that I find really beautiful.
I've been saying this for years. Cancer therapeutics is a population biology problem, not a cell problem. Going after individual cells and individual mutations, it's like playing whack-a-mole. Cancers will always evolve faster than we can come up with new therapeutics. We need a solution that can evolve as well, and that is our adaptive immune system. I believe that we are the fastest team of our size ever to go from therapeutic concept to actual tumor shrinkage. Let me walk you through Karma's past 4 months. 4 months ago, we deep cleaned and rebooted our entire lab and started again from scratch on cancer. A month later, we'd engineered the best non-viral DNA delivery system, achieving 95% transfection efficiency with fantastic viability, and this represented about a 10-fold improvement over current tools. We even showed that our LNPs maintain transfection efficiency even after they've been frozen, unlike current LNPs. And this is all engineered in-house. We haven't licensed any IP at all. But don't just take my word for it. We have partnerships with multi-billion-dollar companies, and they're telling us that we can do things that they've never seen before. Just 2 months later, we'd verified biodistribution to the spleen and showed excellent expression in vivo even 7 days after treatment. This was dose dependent. We also verified that our LNP did indeed transfect macrophages in vivo. It was around this time that we demonstrated that we can co-deliver DNA and RNA together in a single LNP. I can't think of another company that can do that. The implications are that we can engineer cells in vivo for long or short-term effects as needed, and we can even permanently engineer them with gene editing tools. Don't forget though, we are a therapeutics company to our very core, and we've only talked about our tools so far. I want to remind you that our goal is to weaponize and galvanize macrophages so that in vivo, they actually attack the tumor. This unleashes the full power of the adaptive immune system on the cancer, no matter how it evolves to escape. So the question is, does engineering our macrophages have a real therapeutic effect? Well, just a month after we verified splenic targeting in vivo, we were able to shrink solid tumors by 60% in mouse models over about 3 weeks. We're currently working on revalidating and optimizing these results, but we're also testing whether our approach can synergize with other existing immunotherapies that can't penetrate solid tumors. My vision for Karma is to be the best therapeutics company in the world by being the best in vivo cell therapy company in the world. Full stack. Cancer is just the beginning.
Welcome to Alumni Update. I'm your host, Wesley Dang.
And I'm Poe Bronson. Our first update is from our animal-free leather company, MicaWorks, which makes a beautiful luxury product named Rishi. Their South Carolina factory, fully automated, is up and running. Beautiful videos coming out of there. And in the same market sector, IndieBio, which just graduated from IndieBio 9 months ago, is already on the runway in Paris at Fashion Week with Balenciaga with their beautiful new product, Luniform. Over to you, Wes.
Notco, aka the Knot Company, the AI-powered food company, which you may have seen in your grocery stores as Not Milk or Not Burger, and in Shake Shack as Custard, is now following up on its previous partnership with Kraft Heinz with a new partnership with the Mars Company.
Wheat farmers in Argentina suffered a major drought this year, but those who were customers of our company, IndieBio, had quite wonderful results. Congratulations to IndieBio and those farmers. And also in the food sector, Vertical Oceans is moving to Long Beach, California from Singapore. 1063 Therapeutics raised a significant Series A round, led by Hatteras out of North Carolina. 1063 is attacking Max complex with a very unique strategy. Good luck to them.
For those of you looking for alternative meats in the deli and sandwich shops, you may have seen Prime Roots, who just raised over $30 million in their Series B and is now going to scale up to make even more deli meats coming to more stores near you.
This is probably not news to you, but Upside Foods, born originally at IndieBio as Memphis Meats, received USDA approval, first cell-based meat company of its kind, and has announced a large factory in a suburb of Chicago that will be scaling up their meat products. Everyone likes blue jeans and denim jackets. I know you do, I do. And our company Hue, a biomaker of indigo to color all denim in the world sustainably, has just inked a major deal with a denim mill in Asia.
And that's it for Alumni Update.
So for us, it always starts with the founder. You need a founder who's almost obsessed with solving the right problems. And that's kind of what you saw with JP, right, when you met him first?
Here's a scientist from the middle of Mexico, and he was totally impressive with how rigorous, meticulous he was in his science, translating it for decades and explaining it to us simply and clearly in an hour.
Right, right. And he then did this amazing mindset shift. He went from researcher to drug developer. He just wanted to learn the PK, the PD. Week on week, he would come back with data, with the right models to use, and asking us on choice of indication selection, which is crucial here, right?
Absolutely, yeah. And he knocked down those milestones in the program, really like a drug biotech company in the making and a drug CEO in the making.
Right.
And anti-VEGF is a $15 billion market, totally ripe for disruption. Let's go see how he does it.
With age, and for millions of diabetics, the retina, the light-sensitive organ in the back of our eyes, essential for vision, is slowly invaded by dysfunctional blood vessels that leak and proliferate. a process known as angiogenesis. Slowly, then suddenly, patients find themselves living life with literal blind spots in their vision. Some lose their vision entirely. Blocking angiogenesis with antibodies or small molecules by inhibiting the vascular endothelial growth factor, or VEGF, the driver behind angiogenesis. It's a validated therapeutic strategy. These treatments, however, are highly invasive, painful injections going deep into the eye. Despite high prices, treatments remain partially effective. Doing too little, too late. It's no surprise that adherence is so low. At VIAN, we want to see beyond. At the lab of Carmen Clapp in the National University of Mexico, we discovered a natural molecule found in our bodies with essential roles in the inhibition of blood vessel growth in the retina. This was promising, but there was a catch. Producing the natural molecule recombinantly proved really hard. We kept running into problems. Yields were low, folding was a problem, optimization hard. It was a dead end. Understanding this molecule became an obsession for me. I started studying it bioinformatically and ended up analyzing its structure function deeply. Somewhere in this process, this became way more than my PhD thesis. It was as if the problem chose me. I discovered a very short linear motif in this molecule. I observed that only 3 amino acids act as the core structural determinant for an angiogenic function. This very short peptide was easily synthesizable, optimizable, and very active. It was full of potential. Fast forward to today, this research has blossomed into C4551, a first-in-class solution that builds on the peptide we discovered. It is highly potent, stable, and permeable. It can get into the back of the eye in high therapeutic concentrations after a single eye drop. Try doing that with an antibody. Our preclinical in vivo diabetes models show how a daily eye drop of our peptide for 5 days significantly inhibits vascular hyperpermeability in the retina. Napoleon Ferrara, a fabled scientist known for the discovery of VEGF, a discovery that has driven all major drug approvals in this field, is an advisor to this company. His interest and guidance are a vote of confidence that we need to open up a new frontier in this market. Ours is a true disruptor, with the specificity of peptides, matched with the efficacy of antibodies, and surpassing the potency of small molecules. With our low-cost manufacturing, we offer a rare arbitrage. Our eye drops will penetrate this market by undercutting prices without trading off our margins. We see a future where an elderly couple battling age-related macular degeneration shouldn't have to battle the added inconvenience of monthly hospital visits. We see a future where growing incidence of diabetes, especially in emerging economies like Mexico, shouldn't burden patients financially. We see a future beyond the blind spots of today's standard of care.
I think Liliana can be one of the most impactful carbon removal companies there is. They work in the ocean, they leverage its incredible scale, they use the speed and inexpensiveness of biology by picking really precise phytoplankton species that divide incredibly quickly and actually have stored, trillions of tons of CO2 over millennia and eons. What's more than that, they actually matter to customers and are getting pull from different countries because they, solve an immediate problem. They bring back biodiversity, they help fisheries, they remove pollution. So they actually have this, co-benefit that not very many companies can do, and they can do it right now.
I'm Ben Slotnick, founder and CEO of Liliana. We're developing the lowest capex carbon removal technology using Earth's oldest, largest, and most durable carbon sink, phytoplankton. Humanity relies on phytoplankton at a planetary scale. They are responsible for almost all of the oil we've consumed, most rock quarries we mine, the air we breathe, and the carbon that is naturally stored. As biogeochemists and marine biologists, we've figured out how to mimic this process at the speed and scale necessary to remove millions of tons of CO2 in the coming decades. Our society is already creating massive phytoplankton blooms in hundreds of areas around the world. As we unintentionally release millions of tons of nitrogen and phosphorus into the ocean, we create explosions of growth. Just not the right species to reliably and durably sequester carbon. In contrast, we've carefully chosen specific varieties of healthy native diatoms, favorable to marine ecosystems, that naturally sink to the ocean floor and can outcompete harmful algae that create dead zones, such as the Gulf of Mexico, the Baltic Sea, and Chesapeake Bay. We cultivate our diatoms using low-cost photobioreactors we manufacture ourselves. We've developed proprietary dispersal methods to ensure sustained exponential growth, meaning our CapEx and OpEx is incredibly low, targeting under $30 per ton CO2 captured at scale. Surgically dispersing upstream of dead zones, we're positioned to front-run harmful algae, removing excess nutrients. Our diatoms grow fast, sink fast, and preserve well in sediments at the bottom of the ocean floor, providing permanent carbon storage. We know this works from our pilot along the Gulf Coast in 2022. As we dispersed over 1 week, we deployed oxygen monitoring sensors and saw that along our path of release, dissolved oxygen levels increased by over 20%, meaning our diatoms prevented harmful algal growth, which depletes the concentration of dissolved oxygen as they die and decompose. As we prepare for larger deployments, our chief scientist, Michael Beeman, has developed a full-stack MRV system with high reliability to show we can safely scale this technology to help ecosystems and to eventually capture gigatons of CO2.
Our MRV tech stack is based on over 20 years studying marine microbes, dead zones, and biogeochemical cycles. We take direct measurements of key parameters before, during, and after deployment, so we're not reliant on indirect measurements or on models. Then our system is modular and scalable, so meaning that we can work vertically in the whole water column to capture complementary data, and we can scale up and we can spread out over a wider surface area as needed.
We are working with regulators in several key dead zones where we will conduct our next pilots, and have project financing and a carbon credit buyer lined up for one already. Our seed round will enable us to do 60 dispersals, capturing over 50,000 tons of CO2. Please join us as we decarbonize and revitalize our oceans, ensuring a healthy, thriving planet for future generations to come.
Cresco is truly an N of one food as medicine company. There's no other company in the world even pursuing this. And Silvana has a massive head start because she's been studying these dietary RNAs for 2 decades. I think of it as something that could go down into the canon of food of medicine or the sort of hall of fame of food as medicine. They're so powerful and they can already make lots of products off their core library of the dietary RNAs.
Hey, you, citizens of the world. This is for you. Are you taking a trip soon in the coming months? Pay attention.
Citizens is a new kind of supplement company with a library of bioactive ingredients that no other company in the world has or even knows about.
We all love to travel, but it takes its toll. We are going to give you the best travel week you ever had. Sleep at night, energy in the day, an immune boost so you don't catch something. Fat burning and glycemic control for all the great food you eat. We even have a topical supplement to keep your skin hydrated and beautiful.
At Citizens, our bioactives are the results of 2 decades of research in RNA biology. Humans did not evolve to eat processed food or even fresh products shipped thousands of miles over days. We evolved to eat foods off the savanna and to eat them right away. By eating right after picking or squeezing, you benefit from a special class of supernutrients in the cells called dietary RNAs. They are enormously important. But in modern society, These super nutrients degrade by the time we are eating them. Citizens is the first in the world to stabilize them and give you the benefits of nature that has just been picked and squeezed. All our supplements are formulated from foods and flora you already trust to deliver natural health. We have built the world's largest and only functional library of these dietary RNA codes. We have 255 shrubs under study, and we have discovered dozens of very strong benefits. No other supplement has this power. Look at the impact on boosting serotonin and reducing cortisol.
Say goodbye to troubled sleep. Want energy? We got it. Fat burning, glycemic control, sugar uptake in your muscles.
You'll be going all day. Traveling also disturbs your gut microbiome, and you often pick up bad bacteria.
We have the fix. All our products are safe and natural. We hope when you get back home that Citizens becomes part of your daily routine. We will be launching in late 2024. Our cost for 30 pills right now is $9, but we will get that down to $2. Our pricing strategy is in process.
Citizens is a revolution in food as medicine.
Citizens, travel well.
You know, I admit I didn't understand what made carbonate so special when I first met them. They came in saying we could capture carbon, we could utilize that carbon and make green hydrogen at the same time, and I thought it was just a combination play. And it wasn't until we dove into the science that we really understood what made this catalyst so special, that it was precisely the act of electrolyzing that carbon that allowed them to skip the key energetic steps in the capture process. And then they're making hydrogen on the anode side. And we thought that was truly a differentiator in the electrolyzer space.
This is the dream: to take carbon out of the air and turn it into electrofuels, or e-fuels. In the past few years, a lot of climate startups have popped up, claiming to do exactly this. But there's a dirty little secret in all this hype. It takes a lot of energy. Let's dive into where that energy is needed. First, let's get one thing clear. The goal is to make green syngas, which is CO and H2. Once you have these two components, you can make so many things. Second, the goal here is to capture carbon out of the air and use it. But for all the energy needed to draw in the carbon, we need even more energy to release and purify it. It's all a balance of stickiness. The stickier your liquid is to CO2, the easier it is to catch it, but the more energy you need to unstick it so that you can use it. Third, once you finally have the CO2, it takes a lot of energy to rip an oxygen atom from CO2 to make CO. The same goes for making green hydrogen from water. A lot of research has been done on making electrolyzers that either split water or split carbon dioxide. Startups are trying to cobble together these systems to make green syngas, but they're complicated, with lots of parts. And more complexity means more efficiency loss. And there's always some wrench in some part of their system. Expensive catalysts made of gold, iridium, or platinum, super high operating pressures, non-durable membranes, etc. Carbonate has a simpler method. A single-step solution that takes water and air and makes green syngas in a single electrolyzer. Directly, in the correct ratio that customers actually want their syngas, with low energy requirements, with a cheap, durable catalyst. Our research comes from Professor Ronnie Neumann's lab at the Weizmann Institute. His lab discovered a way to capture CO2 and convert it immediately, without having to boil the CO2 just to purify it. Our electrolyzer also splits water into hydrogen on the other side of the membrane, not in a completely separate electrolyzer. Put simply, our chemical reaction can literally skip and combine steps in a way that others cannot. And that is why we can save up to 50% of the cost of making green syngas, which then drops into our industrial partners, who can use our syngas to make everything else. Sustainable aviation fuels, green methanol, chemicals, plastics, etc. They're ready to convert syngas right now. All they need is an economical and scalable source. Just a few months after setting up our own lab this summer, our dedicated highly experienced team has already demonstrated a fully integrated unit that produces green syngas with 90% Faradaic efficiency. Carbonade will be the cheapest feedstock for the carbon utilization market. Because you can't reinvent the supply chain by charging a green premium.
We should be high-fiving.
All right, all right. Well, I hope you guys enjoyed the show. Founders, don't forget, this could be you. Our next batch is starting the end of January, beginning of February next year, and we are checking our emails every day. We're looking at all those applications coming in. Our first investment starts at $525,000, and we are always investing in new, novel technologies that are helping in human health and planetary health.
And again, to all of our friends in the investor community here in the Bay Area, we will be having a live event here in our new offices in Dogpatch. You can come meet the founders. They're all here between 4 and 7 o'clock tonight. We look forward to welcoming you here, and that's a wrap on IndieBio Batch 14.